So today we are going to see about the structure and functions of the cell wall. Especially you know that already we have discussed about the bacterial cell wall. Today we are discussing about the plant cell wall. So in that a cell wall is a rigid layer surrounding the cell next to the plasma membrane. Okay, plasma membrane is present in both plant cell and the animal cell. Over to the plasma membrane, the cell wall is present in case of the plant cells, which provides the structural support as well as the protection, and also it acts as a filtering mechanism. It is not able to allow all the material. Okay, only through the plasmodesmata many materials are moved from one cell to another cell. Okay, so there are canals which are connecting the two cells, which allow the passage of the material, and that is connected to the plasma membrane of one cell to another cell, and it acts as a channel in the cell wall layer. Okay, so in the plant cell wall, so if you see that it provides the rigidity, structural and mechanical support, and it maintains the cell shape, and also when the growth occurs, where the expansion should occur, so the direction is also given, and the architecture of the plant is also decided by the plant cell wall. If you see that there are different types, herbaceous, woody, so that is based on the cell wall development. So in that case, so the architecture of the plant also decided by that. You have seen this slide in the last practical class. Are you able to remember? Everybody have seen. Everybody showed. Sir, I have seen the cell. I have seen the cell with the onion peel. Am I right? So in that, so you have seen the cell, and also the in center the nucleus also you have seen. But what is the rigid layer which are present in that? That is the cell wall. Okay, everybody have seen that. Am I right? Okay, right. Next, so if you see that the cell wall is next to the plasma membrane, and there are different layers in the cell wall, and that protects the cell. As we discussed, the nucleus is not present as such in the center of the cell, since because the vacuoles are making the different sizes and shapes and store the reserve material, so that it pushes the organelles to one side. Okay, and next to that, what is the morphology of the cell wall? What are the layers present in that? The major regions in the cell wall are middle lamella, then primary wall, then secondary wall. Almost 70% of the plants, they have the middle lamella and the primary wall. Whereas on other woody plants, the secondary wall, different layers like S1, S2, S3, like that, they are having different layers of secondary wall in many woody trees. So what are the three regions? Okay, so which one is next to the plasma membrane? In the initial, it is middle lamella. Then while the deposition occurs, the middle lamella expands, the primary wall enters. Okay. Then when the secondary region develops, the middle lamella little bit more, the outermost layer is the middle lamella, and the inner layers are primary wall and the secondary wall layer. Okay, so how? So middle lamella is the outermost layer. It glue that binds to the adjacent cells. Okay. One cell attached with the another cell with the help of the middle lamella. So if you write, one cell is attached to the another cell by the plasma membrane. In the case of the plants, is it wrong or right? It's wrong, isn't it? So if the plasma membrane is present, over that only the middle layer is present. So that middle lamella layer is important for connecting the one cell to another cell. Okay. So what is the composition, chemical composition of that middle lamella? Okay, that is the pectic polysaccharides. Okay. So pectic polysaccharides, pectin, then polysaccharides. You know that it is a glucose containing, that like glucose there are oligosaccharides are present. There are many. Okay, they are present in case of middle lamella. Okay, then next to that the primary wall. So this is mostly present as a rigid layer and deposited in between the middle lamella and the plasma membrane, and during the active growth they are deposited, and then it is making the different sizes in the cells. Okay, the primary wall, what are the composition? There we have seen it is pectic polysaccharides, middle lamella. Whereas what is the composition in the primary wall? So it is the cellulose to the level of 15 to 30%, then hemicellulose 25%, pectic polysaccharides 30%, and the protein 20%. Okay, so listen it because we are going to have the slides also. No problem writing everything. Okay, so just if you see that it is a cellulose, hemicellulose, pectic polysaccharides, and the protein. All the four are present in the primary cell wall. Okay, and that content, whatever what we are saying, 30%, 25%, protein as a 20%, may vary with the various type of plants and age also, because depending upon the deposition it may vary. Okay, so now you know that the plant cell are made up of cellulose and hemicellulose when we study the differences, isn't it? So the primary wall is completely made up of cellulose, hemicellulose, then pectic polysaccharides as well as proteins. So there are good uses of these. We will see it in the future slides. Okay, then polysaccharides in the primary wall, how they are formed. Okay, there are a linkage. So in that, especially the cellulose, it has a linkage of 1,4 beta D glucose residues are linked. At the 1,4 linkage occurs between the glucose subunits, and then it forms a layer of cellulose. Okay, and in the case of the hemicellulose, they are the same, but instead of the beta D glucose residues, here it is hexosyl. Hexo, 6 number, any kind of modified molecules or carbohydrate molecules are present in, sugar molecules are present in that. Okay, the prominent hemicellulose is the xyloglucan. Whereas in the cellulose, which one? Glucose is the main one. There is no other. But here the xyloglucan is the major one. Besides that, you have the other kind of hemicelluloses, that is called arabinoxylon, glucomannan, galactomannan, depending upon the two units, galactose and mannose. Okay, arabinose and the xylose, glucose and mannose. When they combine together, they have the different names. Are you able to understand? Hemicellulose like that. Whereas in the cellulose, glucose. Okay, so there the linkage is 1,4 beta D glucose residues, whereas here it is same beta D linkage but hexosyl residues. Okay, right. So in that case, the third one, pectin. So what are the polysaccharides? How? Here also 1,4 linked, instead of beta D, alpha D galactouranic acid. Okay. So in that case, how to call them? Because the residues are galactouranic acid. So you have to add everywhere it is like a galactouronans. Okay. So in that you can say homogalactouronans or rhamnogalactouronans. Don't bother about the words. You should know galactouranic acid residues are present here in case of the pectins. So we have seen cellulose, hemicellulose, then pectin. Okay, right. So how they are arranged? See this figure for a minute. There is a bottom plasma membrane. Are you able to see that? Which is of dark yellow in color. Dark yellow in color here. Then over that the primary wall is deposition occurs. So in that how it is arranged? You can see the cellulose, a fiber like microfibril or structure united together, and it looks like a stick, isn't it? So that is present in abundance, and they are connected with a molecule of hemicellulose. Okay, and a small fibrous structures which are present as a network, which is the pectin. Is it clear? Different colors, that is, one is the brown, another one is black, and the third one is light green. Are you able to see the all the three? How they are arranged in the primary cell wall? Okay, right. Okay, primary cell walls are extended or they are growing. How it is possible? By a mechanism called acid growth. Acid growth mechanism. So already we have seen in the practical class. So how if you are watering a plant, so they are becoming turgid. Okay, if you are not watering the plant for 2 or 3 days, they are flaccid or they wilt. How? If you are watering the plant, the turgor pressure is maintained, so that makes them to be straight leaves or shoots or whatever it is. So if the water is not there, so the water movement from the cell may come out. Okay, so that it dry, it becomes wilt. Okay, here it has to be making the growth. How? The hydrolysis occurs. How the hydrolysis occurs? Acid hydrolysis occurs. Okay, so we call it as turgor driven and cellulose microfibrils, and the weaker point, that is stronger is cellulose microfibrils, whereas weaker one is hemicellulose and the pectic polysaccharides. They are catalyzed by a protein by the enzyme called expansin. Okay, the expansion occurs, so that proteins are named as expansin. Okay, so that the growth is accommodated. Is it possible to accommodate the growth over a very large extent? No, because this cell to cell interaction is also important. One cell grows in an enormous way, another cell grows in a moderate way, and another cell grows in a very lesser way. So is it possible to maintain that layer of cells? So it is hard. So uniformly the cells are growing to a particular level. Even the acid hydrolysis occurs because of the expansion proteins. Certain growth only get accommodated. After that, what will happen? The primary cell wall is becoming rigid. Then if the accumulation of other substances like lignin or cutin, suberin comes, then it forms a secondary cell wall. Okay, so in the primary cell wall, the growth is the elasticity of that growth, so that only even if you are not watering for 3 days, the plants are able to thrive rewatering during rewatering. Okay, it accommodates the osmolytes which are present in that are making them to give the movements. Okay, but so until up to a certain stage. Okay, just you can see this is a total cell wall microstructure. This will be like that fibrous nature. Okay, either it is cellulose or hemicellulose or pectins, all are mingled together. Okay, and then it looks like in a macro structure, it will be like this cell wall. Okay, so when it comes for the secondary wall, what will happen? So next to the primary wall, additional layers are deposited. Okay. Once the growth is stopped for the plant, but it go on depositing the matter. So in that case, Between the plasma membrane and the primary cell wall, many substances are get deposited. Okay, that layer we are calling it as secondary cell wall. Okay, this is mainly for support. At that time, the cell begins to differentiate. What is the procedure for differentiation? What is the explanation for differentiation? You are getting a lot of cells in our body. How? When they are getting differentiated? Particular function, isn't it? We are saying that kidney cell, we are saying that liver cells. Okay, so like that, the differentiation. Here the root cell, stem cell, flower. Okay, so everything differentiated during these stages. Okay, so the secondary cell wall is also for support, and it comprises of cellulose and lignin. Now you can say that what is the composition of middle lamella? Not pectic polysaccharides. That is in case of primary lamella. Then primary cell wall, cellulose, hemicellulose, pectin, protein. Okay, then when it comes for the secondary wall, it is of cellulose and lignin. Okay, besides lignin, there are some more also, you will see. And so, layer by layer, it is able to be distinguishable during the accommodation of differentiation as S1, S2, S3. It is secondary wall, so they call it as S. Okay, S1, S2, S3, which differ in their orientation as well as their growth depending upon the cellulose microfibrils. Okay, and when they grow, they deposit a new layer, and the oldest layer, it is near the primary wall. Then the newer layer are present below that. So where the newest layer is present? Next to plasma membrane. Okay, so the pectin rich cell wall is laid down between the two daughter cells that cleave during the cell division. Okay, and the younger region of the wall collectively they are laid down successive layer adjacent to the plasma membrane. Is it clear? The middle lamella is the outermost layer, then primary wall, then secondary wall. S1, S2, S3. S3 is nearer to plasma membrane. Okay, right. Then in this secondary wall, there are lot of substances as we studied, mainly the cellulose, then hemicellulose, then lignin we told, and the lignin 10 to 25% it is present, and this lignin is most important one for strengthening the wall, and especially besides the lignin, and silicon is also present in case of the grasses, silicates. Okay, calcium silicate, like that. The silicon uh, elements are present in the cell wall in case of the grasses. So when the herbivore or any kind of insect which feeds on the grass, okay, so they are chewing with the mandible. The larva chew with the mandible. Because of the lignin deposition and the silicon crystals are present in abundance, if they are present in abundance, the mandibles are worn out. Okay, they are not able to chew much. It gives resistance. So we call it as defense mechanism, defense against the herbivore, protected from herbivore because of the silicon presence. Okay. Then the polysaccharides we have seen already, the sugars, building blocks, and the variety of compounds, proteins, lignins, and we will see one by one now. Cellulose, beta 1,4 glucan, and just to see that 25,000 glucose residues are deposited and they are united one with the another in the angle of 1 is here, 4 is here. Are you able to see the hexose molecule, glucose molecule? One of the one glucose molecule is attached to the fourth position of the next glucose molecule. So it is an upside down. So that one unit, one glucose, another glucose joined together in the reverse orientation that portion we are calling it as cellobiose. One glucose with another glucose attachment, it is called cellobiose. Then it forms a hydrogen bonds in many places, intra as well as inter, and it forms a chain of molecules. So it looks like a microfibril. Okay? And these microfibril are united. 36 microfibrils are get united, and it forms, the microfibrils are of 5 to 12 nanometer wide, and it gives the strength. Okay, so have you seen the wire, three-phase wire, or that is used for electrification? Inside that there will be a different color wire. Similarly, here the 36 type of 36 microfibrils are united, and it becomes as a bigger one. So that is the fibrillar structure. Then we will see it in the diagram. Okay, this is in the reverse orientation. Cellulose structure is on the reverse orientation, 1,4 linkage. Just in this diagram, it is very clear. You can see that the first layer, how it is. You see the figure in the first layer, the 1,4 are able to identify on the reverse orientation where the CH2OH is present in one glucose molecule at the top, in another molecules at the bottom. So one of the first position is attached to the fourth position of the next glucose residue. Is it clear? Like that it is go on attaching. Okay, and this position, and because of that it forms a fibril like structure. Yeah, then a small fiber joined together, it forms a larger fiber. Okay, and that is present in case of cellulose, as we say that it is a bigger fibrous structures which are present in as like a brown color in earlier cell wall structure also we have seen. Now are you able to write about cellulose, how they are formed? This is about cellulose. Am I right? Okay, glucose residues, reverse orientation, 1,4 microfibril formation, 36 joined together, and then it forms the cellulose fiber. Okay, so this is the structural arrangement of the cell wall. Same thing, glucose monomer and the glucan chain, microfibril, and the total structure. Okay, then hemicellulose, how they are present. Okay, and as like what we say, it is also a beta 1,4 backbone, but relatively short side chains are present, and also they are soluble in alkali, and common are xyloglucans. Instead of glucose it is xylose and glucose. Here also 1,4 beta 1,4 backbone only here also present, but the that is the sugar molecule is differed. Okay, hexose molecule is differed, various hexo. So glucoarabinoxylans, arabinoxylose is one sugar, glucose is another sugar, they are attached, they are very common, whereas the less commons are glucomannan, galactomannan, leave that. Okay, one example, if you know that, that is enough. Not with glucose alone, you should know one another hexosyl attached with the glucose or galactose, and then they form the cross-linking glycans in case of hemicelluloses. Okay, they don't form microfibril. So what will happen? Microfibrils are formed and they are united, so it gives very good strength in case of primary cellulose. Whereas here in the hemicellulose, it doesn't form a microfibril. Okay, and so the hydrogen bonds also present, but a fucose sugar is on the side chain. So that is about the hemicellulose. Then pectic polysaccharides. As we told, galactourans are making the pectic polysaccharides. So they are pectic acids. Galactourans are the pectic acids. So they are making a polysaccharides, and this is also that pectic acid is extracted from the wall with the hot water or dilute acid or with the EDTA, and they remove the easiest part removed from the cells. They form gels. So everybody, I think, using the jelly sweet, isn't it? That is made up of pectic polysaccharides. Okay. So in that there are so many methods are there. Leave about here they are secreted from the Golgi and then calcium form the cross linkages helical in shape. So this is like that. There are so many rhamnoglobulin etc. Then we will go to the protein. There are three types of important protein, that is the glycoproteins are important. What is the glycoprotein? Anyone? We have studied in the fluid mosaic model. Am I right? In the while studying about the lipid structure, it forms the protein is attached to the carbohydrate. Okay, glycoprotein. Okay, in that there are three types of glycoprotein are important. First one is hydroxyproline rich glycoprotein, HRGP. Hydroxyproline rich glycoprotein. Then proline rich protein. Then glycine rich protein. So what are they? They are all proline, glycine, hydroxyproline or amino acids. They are present in abundance in that particular protein. So we are calling it as the proline rich, glycine rich or hydroxyproline rich. Okay, and these form a rod like structure and they form the different one as a beta pleated structures, so that it is able to form that particular layer. They are able to make it, and the extension protein is used in expansion of the cell wall, and they are well studied hydroxyproline rich protein. In the first group, the expansion is the example. So you can see now all the structures which are present in the cell wall. Either it is cellulose, hemicellulose, pectic polysaccharides, then proteins. Are you able to see that? If you arrange in different fashions, one is dense, one is loose, then we are calling type 1 cell wall, type 2 cell wall. I will show it. Just this is type 1 cell wall. Whatever the components we have seen, this is type 1. Just you observe it. இந்த படத்திற்கும் அந்த படத்திற்கும் 7 வித்தியாசங்கள் உள்ளன. அந்த மாதிரி பாருங்க. இப்போ type 1 cell wall, this is okay. Then type 2 cell wall. Now at this level, you know there are two types of cell wall because of the rearrangements of the various layers. It may be a cellulose or hemicellulose and the pectic polysaccharides and the protein layers. They are in different type of rearrangements, so that they are the plant cell walls are classified into type 1 cell wall and the type 2 cell wall. That's enough. Are you able to appreciate the difference between the 1 and 2? Which one is very loose? Easily, type 2 is very loose. Okay, whereas very fixed one, sorry. What happened? Oh. Okay, so we have seen the type 1 and type 2. Okay, and then secondary cell wall, I think, as we say that the less pectin and the hemicellulose and the lignin is the important one, and three compounds are present for the lignins. They are the coumaryl alcohol, coniferyl alcohol and snapyl alcohol. Okay, and these compounds form the lignin in different plants with different alcohol. In pteridophytes, coniferyl alcohol, whereas in dicot lignin, coniferyl and synapyl alcohol. All groups, only very small amount of coumaryl alcohol is present. Okay. So this is for how the lignin formed. Okay. Now you can see a site of synthesis. We are saying cellulose, hemicellulose, pectins, proteins, everything. Proteins are synthesized in ribosomes. Am I right? Are you able to see that ribosomes? They are saying site of synthesis, rough endoplasmic reticulum, site of synthesis of cell wall proteins. In the last one, you can see that HRGP or PRP, proline-rich protein, glycine-rich protein, like that. The site of synthesis is rough endoplasmic reticulum. Okay, site of synthesis of pectins, where it is? It is a secretion from Golgi apparatus. Okay, so site of synthesis for them is yellow in color. Just you can see that they are the Golgi apparatus. They are the pectins. So it may be rhamnogalactouranin or rhamnoarabinoxylinogalactouranin. Like that, there are so many pectins are formed, but the site of synthesis is Golgi apparatus. Then site of synthesis for cellulose is plasma membrane. Okay, and part of the same hemicellulose also in that stage, in that place only, because both are one and the same except the glucose is the modification, and the callose also forming in that. Okay, the last slide is functions of cell wall. What are the functions of the cell wall? Okay, you can write yourself, it is self-explanatory. First one is cell shape. The shape is decided. There are difference between the animal cell and the plant cell in the shape. Okay, maintaining and determining the cell shape. Then support and mechanical strength because of the cell wall. Then it prevents the cell membrane from bursting in a hypotonic medium. When we study about the osmosis, we will see that. But as I told, it holds certain capacity of turgor pressure. Over that, what will happen? It bursts. Okay, we will see it in clear in the while studying about the movement across the membrane. Then controls and rate of direction of the cell growth. Okay, depending upon the deposition of S1, S2, S3 or primary wall, it makes the cell to grow at a particular direction or not. Then also regulates the cell volume. It allows up to certain volume. Then it is for plant architectural design. Okay, it is a simple cell wall, so it may be of very soft succulent. So if it is of a little bit more with the primary wall, so it is of herbaceous, somewhat woody. And if it is of with the secondary layers in different layers, it is of very much woody. Our teak wood tree is decided by, the price is decided by, in teak wood, hard wood. That hard wood is made up of cell wall, that secondary layer deposition. Okay, how many layers deposited? So that is decided by how many rings are formed in the wood. They will see that while cutting the wood, so how many rings are formed in that. So if it is of more ring, good teak. So that is only, it is in your cart or any wooden material, wooden furniture, you can see a ring like structure which is present in the materials. Okay, that is the secondary wall. Okay, next is the controlling the plant morphogenesis, how they are differentiating. Okay, if there are differentiating, how the primary wall differentiating, and then it forms the secondary wall deposition. Then recognition process. Why it is called recognition process here? Cell wall, how it recognizes? Anyone? We discussed. But how it recognizes? What is the recognizing molecule present in here? In the cell wall? Somebody is, yes, protein. What protein? glycoproteins. Glycoproteins are always signaling molecules. It may be a glycoprotein or glycolipids. Okay, they are in recognition. That is, it recognizes what is coming to the cell. So the receptor is present in the cell. When the external material comes, it accepts, the protein accepts and then take them inside the cell. Okay, so it is recognition. Then important in forming the root nodules and also pollen style interaction. Everybody know what is pollen, what is style, isn't it? That interaction is mediated by this chemistry. Okay, metabolic role is enzymes for transport and secretion. As we say that physical barrier, then carbohydrate storage in seeds, then signaling, as we say that not only recognition, the signaling of particular compound. Okay, that is the role of this various molecule. So in that, one of the signaling molecule is oligosaccharin. You know saccharin? This is oligosaccharin. Okay, so in that, just if you see that it stimulates the ethylene synthesis. Ethylene is good for good. So it is for ripening. Okay, so stimulate the ethylene synthesis. Then induce the phytoalexin. If any pathogen, that is bacteria or fungi comes, okay, during the interaction, there should be an opposite reaction, isn't it, to protect themselves. So for that it induces the phytoalexin synthesis, and also it induces chitinase and other enzyme. Okay, and it also increase the cytoplasmic calcium level. These are all when the pathogen interacts with a plant, the cell wall in that the oligosaccharides are present, they stimulate these kind of responses. Okay, because of that it causes an oxidative burst. Okay, so then the pathogen are not able to enter inside. It is localized to particular region. Okay, so by that they give the defense. So one of the example is Phytophthora, that is the, it forms a this is a pathogen and the host plant, how they are making. Then what are the economic products we are getting from the cell wall? Now you can tell, we are getting paper. Where is the paper mill is present near to Coimbatore? Anyone? I think all are from Tamil Nadu, am I right? Only very few. Okay. So where the paper factories are, paper mills are present? Karur, then Pugalur, am I right? Near Karur. Puliyur or Pugalur? Pugalur. Then next. Here near Erode there is another one. Okay. So then near Tirunelveli, you know that there is a way by near Ambasamudram on their way, sun paper mills. Okay, so like that there are so many paper mills. What is the material for paper mills? The wood, eucalyptus, casuarina, sauku, thailamaram. So அதை வந்து பீலா ஆக்கி தான் அதுக்கு பேப்பர் தயாரிக்கிறதுக்கு யூஸ் பண்றாங்க. So that only, so if you are not wasting the paper, you are saving the trees. That is the slogan, isn't it? So you should know what is that. Then paper, then wood, firewood or any kind of wood use, you know, that is for cell wall rigidity. Then fiber. So we have to take fibrous food, otherwise constipation will come. Okay, so fibrous food, fiber rich food. Okay, not carbohydrate rich food. Fiber rich food we have to take it so that it is good. Fiber, energy, shelter. So everything, total roughage of our diet. நம்ம வயிறு அடைக்கணும்னு சாப்பிட்றோம் இல்லையா, வெறும் கொஞ்சமாக சாப்பிட்டோம்னா என்ன ஆகும், பசிக்கும், இன்னும் கொஞ்ச நாள் இல்லையா பசிக்கும். ஸோ அதனால் என்ன ஆகும், மாடு என்ன பண்ணும், வைக்கப்புல்ல சாப்பிடும். வைக்கப்புல்ல என்ன அப்படியே போய் இது பண்ணிக்கிட்டு இருக்கும், அதுக்கப்புறம் அதை ரூமினேட் பண்ணிக்கிட்டே இருக்கும். ஸோ அதுக்கு அந்த மெக்கானிசம் இருக்குது, திருப்பி எடுத்து அதை கொஞ்சம் சின்ன சின்னதாக ஆக்கி திருப்பி உள்ள தரும். ஓகே, நம்மளுக்கு அப்படி கிடையாது. நம்மளுக்கு வந்து சாப்பிடுவோம், பட் அந்த ரஃபேஜ் வந்து கொஞ்சம் போய் அடங்கணும், பசி அடங்கணுன்றதுக்காக கொஞ்சம் கூடுதலாக தயிர் சாதமோ இல்லை எந்த சாதமோ கொஞ்சம் நல்லாவே சாப்பிட்றோம், ரஃபேஜ். ஓகே, ஸோ த ஃபைபர் ரிச் ஃபுட் இஃப் யூ ஆர் டேக்கிங், தட் இஸ் குட் for satiation as well as for all kind of health, good health. Okay, then seed hairs, that is also from cell wall. What is that? What you are wearing the cloth, it is from cotton. Okay, the fiber, that fiber is from cell walls of the plant. Okay, then silk cotton. What is silk cotton? It's a mixture of silk and cotton. Anybody? I think you might have studied in +2. Silk cotton. What is a silk cotton tree? எந்த தலையணை வச்சு படுத்து தூங்குறீங்க? எல்லாம் ஃபோம் தானே? இலவம்பஞ்சு இருக்கு இல்லையா? That is silk cotton. Okay, but now you are having, everybody is having the pillow as made from synthetic one, isn't it? Okay, so that is a silk cotton is that fiber is also useful for various cases. Then stem fibers, that is the bast fibers. Stem is used for taking the hairs. Okay, what are the stem? So it may be a pulichakira mari. Okay, have you heard about cannabis? Okay, then conchorus, then linum, all kind of things which is from stem. Okay, then another one is leaf fibers. Banana fiber is used for now even making our textile products. Okay, there are various products banana fibers are useful. Then agave fibers, there are so many. Agave, sothu kattalam kelivittirukingla, kattala. Okay, so that fiber, that is from leaves. Okay, then aloe vera is not a cell wall product, but that is also from a succulent, and inside that the material is not fibrous one, that we should not say. Then there is a compound used in molecular biology that is called agarose. Okay, just to see the structure, it is from a cell wall structure. Okay, a fibrous material or a gel like substances are formed. Through the gel we are running the DNA or RNA or protein, and that gel making we are using the agarose. That agarose is from the product of the cell wall from various things. Okay, not only in the plant, then marine algae also are used for this. These are the then the between the cells connection point is between the cell connection point is the channels. What are the channels? Plasmodesmata. Okay, desmatum is the similar one. Okay, with this we'll finish, and this formation is in different type. This is not for you, and here there are different types of organization of the in different models.